rheumatic fever and rheumatic heart disease is still being important in several countries. And in Brazil, it is important in some regions. So the disease follow a Streptococcus pyogenes infections, and those people that have some genetic markers that are susceptible to develop the disease will develop different types of manifestation. It starts by a polyarthritis in 90% of children, and it can develop skin lesions as erythema marginatum and subcutaneous nodules, as you can see in also, and the sidereal chorea that makes involuntary movements, and the most severe sequel that is the rheumatic heart disease that leads to valve damage, and these patients had a very difficult life and need surgery sometimes more than once to correct the lesions, and some of these patients are very young children, sometimes being operated 5 or 10 years old. So, here is to give you an idea. These data are from 2 years ago. We, in São Paulo, that is a big city in Latin America, we have the clinical hospital that is a very big hospital, and many people come from all Brazil and Latin America to be treated there. So we have treatment for rheumatic fever, the first time that this patient come. And as you can see, that when they come to the hospital, because they arrive, they have had already the heart damage, most of the patients, and you can see here. So 63% of patients had already mitral valve involvement, and showing that it is a very bad very bad picture, if I can say. And this to give you an idea, the numbers of cardiac surgery that was performed in Brazil in the 18th 2004, it was more than 5,000 surgeries, and it represents around 50% of the cardiac surgeries performed in the Heart Institute. And we have until now around 600 outpatients monthly. 2,000 patients went for valvular surgery, and around 38% of these surgeries are in young patients. So these other slides show also that the data more recently that this surgery represent 70% of surgical cases of valvular disease. Here, just focus on valvular disease, and have the incidence at school age is 1 to 7 and 1,000 children in Brazil, compared with the United States that they have 0.1 to 0.5 in 1,000 in the United States. And it's because the higher incidence of valvular disease in young people. Then, as I mentioned, I will not enter in details of the genetic susceptibility because you have no time, but there are a lot of genes that are nowadays known that play a role in the development of the disease. The classical HLA class 2 alleles defined several years ago by serology and molecular biology. So there are some alleles like HLA DR7 and DR4 that are the most consistent associated with disease, and then with the development of new techniques, several genes were defined by molecular biology, and most of these genes are related with the innate immune response. So that is the first line of the immune response. As we can see here, we have I don't know if it's, I can. So here, these are related to the beginning of the infection, so in the recognition of the bacteria and the activation of complement and other genes with the immune response, and all the cytokines, the inflammatory cytokines, and the regulatory cytokines that, as I show you, has a little number of cells producing regulatory cells in the development of the disease. So, as I told you, after the colonization of throat by Streptococcus pyogenes, there are the activation of the immune system, and as we can see here, activating the bacteria was processed by macrophages, and several peptides of the bacteria are present to this CD4, many to the CD4 cells, and then activate the antibody response. And here we will have the Streptococcus prime, the CD4 cells, that in the periphery blood of these patients will be able to recognize some peptides of the M protein of the Streptococcus, that is the major protein of the bacteria, and this immune response is related with presented mainly by HLA class 2 DR7 and DR53 alleles that are the most associated with the development of the disease. And one data that is very, very interesting is that these people, when they have these few weeks after the infections, they, in the blood, the peripheral blood, they have already cells that can recognize heart tissue-derived proteins. As we can see here, proteins derived from the myocardium and also from the valve aortic proteins. So what happens then? So this, it means that these peripheral cells, primary peripheral cells, go to the heart, and here we can see this is a mitral valve from a patient that was operated and needed to replace the valve. And so this is the typical lesions in the valve, that is the vegetation. And in this tissue, there are a lot of infiltrating T cells. As we can see here, these cells grow in the, only in the presence of IL-2 very quickly when these patients are operated in near to acute phase of the disease. And then we evaluated these cells by cloning these cells, and we could show that in the heart, in the absence of the bacteria, these cells were able to recognize a peptide from the M protein in this region that was an immunodominant peptide, and they were able to recognize some proteins from the valves, as you can see here. And one thing that was very interesting, that patients that were, normally these patients are operating in a chronic phase. Even being operated in a chronic phase, they present around 20 to 30% of autoreactive cells. And sometimes these patients are operating in acute phase. And when they are operating in acute phase, we can have until around 70% of autoreactive cells, showing that during the acute phase, these cells are, for those that are in a second episode, they are reactivating, a large numbers are responsible for the lesions that we can see here, and insufficiency of the valve. And more recently, we could identify some of the proteins that are target of these T cell, Streptococcus prime the T cells. These proteins are mainly vimentin, the collagen type 6, and another protein that are important from the structure of the valve. Another side that was also very interesting, that as we can see here, there are a lot of cells infiltrating the myocardium, as we can see here, mononuclear cells, and in the myocardium and in the valve, also a lot of cells infiltrate. When we look at the cytokines that these cells are producing, we have cells producing inflammatory cytokines like TNF-alpha, interferon gamma, and IL-17, IL-23. And among the regulatory cytokines, we have IL-10 that was produced, but we have only few cells that were able to produce IL-4 in the valve. So this makes some difference for the progression of the lesions, because as we can see here, we have the opposite picture showing that in the myocardium, in which we have the regulatory cell number, normal number, I can say, of regulatory cells, the myocardium heal. And the valve, we have only few cells producing regulatory cells that makes the worsening of the valve and the permanent valvular damage on these patients. So, taking all this in consideration, it was a very difficult task to produce a vaccine against Streptococcus pyogenes without triggering lesions in the heart. And for this reason, we choose some peptides located in this part of the M protein, that is the C2 and C3 block, that is conserved among all the strains of Strep. Nowadays, more than 200 strains now. And this part is so conserved and not trigger autoimmune disease. So, by analyzing a large panel of cells, cells in serum, we could identify two epitopes that we call the T epitope and the B epitope, because these peptides were recognized by both T cells and B cells. And when we compare these two epitopes, we verify that they have a core identical due to the amino acid sequences of the M protein that are in repeat some amino acid residues. So then we decided to design a vaccine composed by 55 amino acid residues, taking into consideration both epitope, as we can see here, the T epitope and the B epitope here, and linked by these 8 amino acid residues as it is in the natural protein. And then we verify by nuclear magnetic resonance how this protein fits. And then as you can see here, we have these different colors, are different models that was studied. And in fact, we have a region that is in alpha helical conformation and some regions that is more not in alpha helical conformation. This is very, very important for the recognition by the immune system, because as it is a long peptide, it will be processed by antigen processed cells and will generate different peptides, and this peptide will be recognized by, will be present by the antigen present cells, recognized by T and B cells in the context of HLA class II molecules. So what you did, so as here is a schematic how we look the peptide. So this is the peptide. This is the different possibility of being recognized, taking consideration how this will fit in the macrophage and in the pockets of the macrophage that needs specific amino acids residues in order to be recognized by the different HLA class II molecules. And so we have 7 possibilities. Consider the peptide, 7 possibilities here represented from A to G of being processed and in order to be recognized by any HLA class II molecule. And in fact, then when we look at the immune response in a very large panel of cells, 250 samples, and we could verify that we have among the different 14 overlapping peptides can be recognized by any HLA class II molecules, as you can see here by the first molecules, and by the second molecules also we have the recognition of all these molecules, showing that this peptide can be a universal vaccine. So then, in parallel, we did a lot of experimental assays in different models of mice. Here, BALB/c, Swiss HLA class, and we also work with HLA class II transgenic mice that it will be more similar than humans, and we also immunize mini pigs that with 25 or 30 kilos that it was like a children. Here is just to show you that streptococcus induce high in a specific IgD antibodies, as we can see here. And the molecule was also recognized in the context of a recombinant M1 protein, and we have no cross-reaction with cardiac myosin. We did also a challenge. This was a challenge for us because it's very difficult to induce some type of disease in the mice. There are no model of the disease, animal model for rheumatic fever. And what we could induce was a peritoneal infection in these animals, and then we followed for 30 days, and we observed a survival of 87% in those immunized mice compared with the controls. Another important property of the vaccine that it induces in different types of mice. We could induce antibodies, neutralizing antibodies. As you can see here, this is infections in HEP2 cells, mimicking a throat infection. And when this was treated with the serum of these immunized mice, we show that we have no colonization, showing that these animals are in some way protected to the infection. Another interesting study done with other set of BALB/c immunized mice is that we observe these data from cytometry analysis. So we observe that the animals that were immunized with streptococcus present neutralizing antibodies. Here is the bacteria, different types of the bacteria. These grow, and the animals that were immunized so have a neutralizing antibodies. And you can see here we tested in 5 different types of Streptococcus pyogenes. This also was very, very interesting assay. We look for the capacity, opsonize the produce antibodies to induce the opsonic phagocytosis, and we also have a very good results showing that as you can see, different types of bacteria again, and the serum from these the immunized mice could induce the opsonic phagocytosis. Just to show you the cells from the peritoneal cavity, here we have a lot of bacteria inside the cells, and mediated by the anti-Streptococcus antibodies. In HLA class 2 transgenic mice, we also evaluate the titers of antibodies, and we have a very high titers, as you can see here, different against different isotypes of IgG. As in humans, these animals recognize all the different peptides. As you can see here, the serum recognizes different peptides of the Streptococcus, and this is a negative result that we showing that these animals did not, the antibodies against the Streptococcus did not recognize any valve protein. So this is a safety assay. And here we follow these animals by 1 year of vaccination, and then we look for the different organs in the tails of the heart. Here is the 4 chambers of the heart. Then we have the valves, tricuspid, mitral, and aortic valves. We look also the articulations, kidney, spleen, brain, and liver, with and no bad reactions or autoimmune reactions were observed. And another very interesting result, that when we are looking from these intralesional cells that we could develop from patients, we found that some of these intralesional cells were able to recognize some peptide of the vaccines. And in the first moment, we think that it could be a very bad answer. But in fact, these the cells that recognize produce IL-10, then we ask if these could be a regulatory T cells. And in fact, we performed other tests, and we confirmed that the Streptococcus has the potential to induce regulatory cells, I think I have here picture. As we can see here is healthy individuals, how the numbers of regulatory cells that these normal individuals have. This is the rheumatic heart disease patients that have a low numbers of regulatory cells. For this reason, they develop autoimmune reactions. And when these cells are stimulated with Streptococcus, these numbers became to the normal pattern of individuals, showing that probably this vaccine will also have a therapeutic effect for those patients that are already sick. The things that is interesting, that this property of induced T regulatory cells is dose dependent. So different from the vaccine that we have a good immune response with 10 micrograms. For regulatory cells, we need 5 times more. So it is interesting, and we are now doing other experiments to confirm the regulatory potential of the peptide, and maybe it will be possible to treat the patients and bring them to a better life. And this is just to summarize. So more or less I talk about that we can have a protection with the vaccine, and we can also induce the therapeutic effect. And now I'm just a few minutes, and now we will start the clinical phase 1. So the design of the study is a random double-blind control with placebo, and we intend to use 3 different doses, and with a 20 days interval and a booster of 6 months. And it will be done in healthy volunteer, aging 18 to 45 years old, to be sure that we will not have troubles with the age. And so we have, now we have already the GMP production. This is being formulated by Instituto Butantan in Brazil, that is a vaccine developer. And we started with the documents for register this, and it is a very long way. Thank you very much, and sorry for the delay. This is the all people that are involved from the hospital, Heart Institute, and is the Heart Institute in São Paulo. Thank you.
Very interesting study. I think if you have any questions, you can discuss with us.
Okay.
Another thing is we'll chat on a big problem for the next